Stanford University.
I wanted to just start by giving a little brief introduction into, oops, went too fast, into the clinical aspects of how we treat sarcomas in the clinic, and particularly focus on two types of bone sarcomas, osteosarcoma, which is the one that most of you have heard of, and then a more unusual pediatric tumor called Ewing sarcoma. And I'll try to link the two together and talk a little bit at the end about why I think the two are related. So just as this keeps going too fast, I don't know why it's doing that. Just a brief introduction for those of you who are not that familiar with pediatric cancers. The distribution of pediatric cancers is actually very distinct than what people are used to thinking of in adult cancers. So whereas adult cancers are mostly carcinomas, pediatric cancers are mostly leukemias, brain tumors, and sarcomas. So we very infrequently see an epithelial-derived carcinoma in a pediatric patient. So leukemias are about a third, brain tumors are about 20%, and then different types of sarcomas represent the remainder, although we do see also a significant number of lymphomas. So I'm going to focus primarily on sarcomas, and as I mentioned, two types of bone sarcomas. So the two types of sarcomas that we work on in the laboratory are osteosarcoma and Ewing sarcoma. And these are really diseases of young patients. So they are seen in adults, as you can see from this slide. But we see most of them in pediatric patients, sort of adolescents being the primary age group. So osteosarcoma and Ewing sarcoma are the two most common types of bone cancers. Chondrosarcomas are very rare, and others we occasionally see. So I wanted to point out that the biology of these bone sarcomas is actually very unique. Many sarcomas, as many pediatric cancers in general, are characterized by translocations. So you heard this morning about from Linda Boxer about translocations in leukemia, and those are very well known. Perhaps less well known is the fact that many pediatric sarcomas and also some adult sarcomas are characterized by translocations. So in the laboratory, we're very interested in these EWS translocations, and that's sort of the classic tumor that's caused by EWS translocations is Ewing sarcoma, which is caused by an EWS FLI1 translocation. But as you can see from this list, there are many other sarcomas that are also induced by translocations. Actually, this doesn't probably project very well, but Ewing sarcoma is one group. Rhabdomyosarcomas are another group of cancers, pediatric cancers caused by translocations, as well as some other rare tumors. But the exception actually is osteosarcoma, where there is no known translocation, and the initiating oncogenic event is not well known. Although some cases are known to be caused by germline mutations in the RB locus. It's still not clear what the initiating event is in most osteosarcomas. So for both Ewing sarcoma and osteosarcoma, there are well-known clinical predictors of prognosis. The clearest one for which there's the best data is incomplete local control. So if you have either a tumor that presents with metastasis at diagnosis, or a tumor that because of its location cannot be fully resected, for example, pelvic lesions have generally poor local control and a much worse prognosis. So, and related to that is metastasis. Again, location. So again, related to the issue of local control. So if you have a difficult location where it's difficult to surgically extract a tumor, that makes it much more high risk. And then an issue that I'll come back to several times during my talk is the question of percent tumor necrosis after chemotherapy, because I think that is an interesting, unique aspect of these sarcomas, which presents some interesting opportunities for biological studies and translational research, and also the size of the primary tumor, again, probably related to local control. So there has been, as opposed to some of the talks you heard earlier, describing the slow incremental improvements in treatment of some adult carcinomas. There's actually been some fairly dramatic improvements in survival in osteosarcomas and bone sarcomas in general, pediatric sarcomas, over the last 30 to 40 years. So in the pre-chemotherapy era, survival rates were very dismal, with only about 20% of patients surviving with surgical resection alone. And in the 60s, it was found that methotrexate was very effective for treating osteosarcoma, and that significantly improved the survival. And then with modern chemotherapy regimens using multiple chemotherapeutics, survival is now, for non-metastatic patients, is now in the 60 to 70% range for both. This is for osteosarcoma, but similar results have been found with Ewing sarcoma. So some of the other sort of genetic risk factors that are important. So osteosarcoma, history of prior radiation. So the classic example of a retinoblastoma patient who receives radiation for local control of a retinoblastoma. Those patients are at very high risk for osteosarcoma, but actually any patient who's received high doses of radiation. Patients with Lee-Fraumeni syndrome are also at risk for osteosarcoma, and this rare syndrome called Rothman-Thompson syndrome. I put here race, which was pointed out to me is not really a risk factor, obviously, but it's just an epidemiological fact that patients who African American patients have a much lower incidence of Ewing sarcoma than Caucasians, and the reasons for that are not entirely clear. So the treatment, just for those of you who are perhaps not familiar with how we treat these diseases, for both Ewing sarcoma and osteosarcoma, obviously diagnosis followed by chemotherapy, followed by local control and more chemotherapy. And again, this is a somewhat unique schema for treatment that, as I'll show you at the end of my talk, prevents some unique opportunities for procurement of tissue samples and analysis of chemotherapy response. For osteosarcoma, we usually treat with high-dose methotrexate combined with cisplatin and adriamycin. The treatment for Ewing sarcoma is somewhat different. It's usually vincristine, adriamycin, cyclophosphamide, alternating with ifosfamide etoposide. But again, the schema is the same: chemotherapy, local control, followed by more chemotherapy. So I wanted to just touch on this issue of percent necrosis. So both for Ewing sarcoma and osteosarcoma, and this is for Ewing's, there's a very clear correlation between the response to the initial, we call neoadjuvant therapy, after the initial diagnosis before local control. If at the time of local control, the pathologist will look at the remaining tissue, and depending on what the percent necrosis is, that's a very strong predictor of how that patient is going to do long term. So patients who have greater than 99% necrosis, perhaps not surprisingly, are going to do much better than patients who have less than 90% necrosis. This is after the initial first 3 cycles of chemo. And that's also true for osteosarcoma. So as I mentioned, that there's been a significant success over the last 20 or 30 years in incorporating these chemotherapeutic regimens for treatment of Ewing sarcoma and osteosarcoma, but there's still a lot that we can do. So one issue is improved survival for localized control with less intensive therapy. So one issue that is perhaps plays more of a role in thinking about these pediatric cancers than perhaps it does in adult cancers is the issue of, well, there's going to be a very long survival of these patients. So you end up thinking about a lot of issues having to do with long-term survivorship and the consequences of having treated pediatric patients, for example, with high doses of alkylating agents, which cause sterility and other issues. So we'd like to be able to treat patients with localized disease with less intensive therapy. And we also have a huge need for improved therapies for patients that present with metastasis, because, as I mentioned earlier, those patients have a much lower survival rate. So that brings us to really the focus of the rest of the talk, which is some various efforts that have been made over the last few years to find molecularly targeted therapies for Ewing sarcoma and osteosarcoma. I'm going to start by talking about some of the clinical trials that have been recently carried out or are being carried out now, and then move on to sort of more laboratory-based investigations, some from our own lab, sort of trying to come up with the next generation of potential therapies. So some things that are currently being tried, I'm going to briefly touch on for osteosarcoma, MTP, which is a muramyl tripeptidase, which is a component of the BCG vaccine, which is a tuberculosis vaccine, which has been found to be to stimulate macrophages in the lung, and there was some preliminary data that I'm going to discuss that led to a trial, which has had some interesting findings, which I think are instructive of some of the pitfalls of doing clinical studies based on biological information. And then we'll talk a little bit about a current trial that's going on using interferon for osteosarcoma, and some preliminary studies using the inhibitors of the IGFR1, insulin-like growth factor receptor 1 for Ewing sarcoma. So this INT0133 was a study that's now closed, where the goal was to look at the combination of ifosfamide and or muramyl tripeptide for the treatment of osteosarcoma. So the standard treatment at the time was this MAP, which is methotrexate combined with adriamycin and cisplatin, and the idea was to look at the addition of both ifosfamide and MTP to improve the outcome. And the basis for this was that for ifosfamide, there had been some phase 2 studies showing some response, a couple of different studies going back a few years, and then also an upfront window approach in metastatic patients that had also shown some responses. So there was some clinical data suggesting that ifosfamide might improve MAP, the outcomes with MAP. With MTP, there was some data. So one of the interesting things about osteosarcoma is that one of the interesting preclinical models for studying osteosarcoma are dogs. So large dogs tend to develop osteosarcoma spontaneously. So they've been used as a preclinical model to develop new drugs for osteosarcoma. So this is an example of a dog study where they looked at this liposomal MTP, which is this BCG component of the BCG vaccine, which is thought to stimulate macrophages and have anti-tumor effects. And you can see here that empty liposomes versus liposomal MTP, there was a clear influence on proportion of patients of dogs in remission. In this case, the patients are dogs. So this was the data suggesting that it might work. And there was also some human data from using historical controls suggesting that patients who received MTP actually did significantly better. So this led to this trial, which had kind of an interesting design that I wanted to talk about briefly. Oops. So it's this something called a 2x2 factorial design. So the idea was we have the standard therapy, which is MAP, cisplatin, doxorubicin, hydrous methotrexate, or we're going to combine that with this MTP drug. So that's A versus A plus is standard therapy or standard therapy plus MTP. And in the B arm, it was either standard therapy plus ifosfamide or standard therapy plus ifosfamide plus MTP. So basically, you have these four groups, and this is why it's called the 2x2 factorial design. And the key point here is that this only works if you assume that there's no interaction between the interventions. So if there's any kind of interaction between ifosfamide and MTP, or plus or minus, then the statistical significance of these studies doesn't really hold true. And one issue that I should mention also is that one of the issues in general with doing clinical studies in pediatric sarcomas is that pediatric sarcomas are rare diseases, fortunately. So the numbers are quite small compared to what you might, where people are used to thinking of, clinical trials with lung cancer or pancreatic cancer, where you have thousands of patients. I mean, in a pediatric trial for osteosarcoma, combining many, many different centers, you'd be lucky to have a few hundred or maybe 1,000 patients. So the interesting aspect here that I wanted to just touch on is that these are the results of this trial, and it's very interesting. So what you see is that if you use the B arm, so basically ifosfamide with no MTP, you actually have a worse outcome. If you have the B arm plus MTP, you do better than the standard arm with or without MTP. So this basically tells you that there was some unanticipated interaction between these four arms, which makes it very difficult to to interpret the results of the study, and it's become a very controversial issue in this field. Why exactly is it that MTP only works in the context of ifosfamide and not in the context of the standard regimen? But basically, to sort of summarize that very quickly, it seems like there's perhaps some biological reasons why ifosfamide stimulates pathways that actually make the therapy not so good, but if you add MTP, then that makes it a little bit better. But in essence, it turns out that it's probably not going to be a very effective treatment. So as I was mentioning before, pediatric osteosarcoma is a rare disease. So one of the things that is sort of unique to pediatric oncology is well, it's not unique. It's done in adult patients as well, but there's a long history of this in pediatric cancer, of these cooperative groups that have now started out nationally, but now have extended to include many European groups as well. So there's this Uramus Consortium, which is a combination of European and North American groups to develop new trials for osteosarcoma and thereby increase the number of patients. So just to walk through this quickly, the current Uramus trial is basically evaluating a couple of things, which is basically the addition of ifosfamide for patients. So basically, everybody gets the same treatment initially, and then they're diagnosed in terms of their response. As I mentioned before, percent necrosis at the time of diagnosis, at the time of surgical resection is a very strong predictor. So patients who have a good response get, which is basically defined as greater than 95% necrosis, get the standard MAP regimen or MAP regimen plus interferon, and this is based on some preclinical data from the Scandinavian group suggesting that interferon has some effects in vitro against osteosarcoma. And then the patients with a poor response initially get MAP plus ifosfamide or with or without interferon. So that's sort of an ongoing trial. And I wanted to touch back again on this issue of using dogs. So this is from a review. It was recently published in Nature Reviews Cancer, just kind of sort of outlining this idea that you could combine clinical trials in dogs with clinical trials in humans to try to accelerate discovery of novel biological therapies that might actually be effective for both species. So that's something that's being pursued more these days, and this is one example of that. So this is a trial again in dogs using mTOR inhibitors against osteosarcoma, with the idea of defining a maximally tolerated dose and doing a lot of the PD studies in dogs, and then more quickly being able to transfer those studies to human patients. I think I'm going to skip over this because I might run out of time, but just briefly mention that some of the other things that have been being looked at. You heard this morning about the HER2. There's some evidence that the HER2 expression correlates with outcome in osteosarcoma, not so well in Ewing sarcoma, but in osteo it does seem like there's a correlation. So the patients that have a negative for HER2 actually have a better survival than patients that are positive. So there's another trial going on now, adding a HER2 inhibitor for patients that are positive for expression of HER2. So that's ongoing, and I'll skip this. For Ewing sarcoma, there's some interesting biology involving the insulin-like growth factor receptor, which is a tyrosine kinase, which is ubiquitously expressed in Ewing's tumors. And there's some preclinical data suggesting that, at least in mouse fibroblasts, when you add the EWS-FLI translocation, expression of the IGF-1 receptor is required for the ability of EWS-FLI to act as an oncogene in those cells. So that's been used as to suggest that perhaps the mechanism might be that the IGF-BP3, which binds to the IGF-1 ligand and is basically inhibits it from acting to the IGF-1 receptor, that EWS-FLI1 maybe inhibits this interaction and therefore potentiates the ability of IGF-1 to bind to its receptor and activate downstream targets. So perhaps blocking this this interaction may be an anti-Ewing sarcoma therapy. So there's a phase 2 study that was recently reported. Actually, the phase 2 study is currently going on. It was started in 2007, and that was based on results of a phase 1 study showing some durable partial responses in patients with Ewing sarcoma treated with this anti-IGF-1 antibody. So that's an ongoing study. So kind of to transition, this is a totally different approach to kind of a novel concept. The idea being here that if you, particularly with these tumors where you have a translocation event that you think is the initiating oncogenic event, if you could identify, you could block the downstream targets of that translocation, perhaps you could find novel therapies for these cancers. And so this is a study by someone at the Dana-Farber Cancer Institute, where they use a sort of a novel approach. The idea is if there's an expression profile of this translocation, in this case EWS-FLI translocation, you can uncover that translocation by knocking down EWS-FLI1 in cell lines and basically uncover this sort of on-off gene expression profile. And if you have some kind of a high throughput way to screen for this on-off state, you could identify novel drugs that would be inhibitors of EWS-FLI1. So basically what they did is they identified this on-off signature by knocking down EWS-FLI in cell lines, and then using this machine called the Luminex machine, which can basically do kind of like a high throughput TACMAN, similar to the TACMAN assay. So you can do, except that you can do like 40 or 50 genes in one well. So it's kind of like a mini microarray. You can basically do a high throughput screen to try to identify compounds that will recapitulate the off effect of the shRNA. And since we think that the EWS-FLI is the initiating oncogenic event, you might be able to identify novel therapies this way. So by doing this type of what's called a GEHTS screen, or a gene expression-based high throughput screen, they were able to identify cytarabine, which is kind of an interesting idea. So cytarabine is a drug that is frequently used to treat leukemias, but had never been thought to be actually effective in Ewing sarcoma. And they found cytarabine to be a hit in this type of a screen. So as you can see here, you have the RNAi control. These are the genes that are off. These are genes that are on. When you knock down EWS-FLI, it kind of reverses the signature, and you get a similar effect with cytarabine. So how does cytarabine work? So it doesn't seem to affect the expression of the EWS-FLI transcripts, but for reasons that are unclear, it seems to decrease the protein level of the EWS-FLI transcript. So that may be how it functions. So there's some ongoing studies now based on these observations in a phase 1 study to look at using Ara-C for treatment of Ewing sarcoma. So to move on a little bit, obviously there's a lot of interest these days in using genomic predictors to study, to identify basically genomic profiles, to study different kinds of cancers, and certainly that's been done in Ewing's and osteo. So this is just from a recent study, very large data set collected at the NCI, looking at many different types of sarcomas, and similar to what people have found with leukemias and with other cancers, not surprisingly, there's a very clear, tight clustering based on the histology of these different tumor types. So osteosarcomas cluster together, Ewing sarcomas cluster together, adult sarcomas clustered separately. One of the interesting things is actually that the pediatric sarcomas, which are mainly induced by translocations, tend to cluster separately from the adult sarcomas, which are mostly not translocation induced. So one of the interesting things come out of that study is that there's a significant specificity to the tyrosine kinases that are activated in specific tumors. So for example, in Ewing sarcoma, those were characterized by expression, at least, of this JAK1 tyrosine kinase. GIST obviously is well known, have overexpression of KIT, and some other ones were found here. So these are potential therapeutic targets that are currently being pursued, at least in preclinical studies. And this is, again, getting back to this idea that because we have access to both the initial diagnostic sample and the post-chemotherapy sample, which is sort of unique in not that many cancers where you actually have easy access to that, and you can look to see which patients responded or didn't respond to the initial chemotherapy based on percent necrosis. This is a study where they looked at the initial diagnostic sample and then looked at how expression of those correlated with whether the patient responded, had a high percent necrosis at the surgical resection or poor percent necrosis, and clearly saw some genes that went up and down. So again, these are genes that may correlate with chemotherapy response. So this is an interesting avenue for further investigation to try to potentially identify markers of patients who are likely not to do well with standard chemotherapy. So that could lead to the design of novel trials, or potentially identify genes that could be inhibited through chemical inhibitors to turn a poor responder into a good responder. So in the last part of my talk, I'd like to turn to some of the things that we're interested in the laboratory, which is really the link between potential cell of origin of these sarcomas and whether that has anything to do with or whether these cancers actually have cancer stem cells or within them, and maybe by studying the cell of origin of the cancers, we can try to learn something about what might be the self-renewal pathways that are being utilized by the cancer stem cells. The cell of origin of these tumors is really unknown. So obviously there are some type of mesenchymal origin because they're sarcomas and they have that appearance. But the mesenchymal lineage, unlike the hematopoietic lineage, which has been beautifully delineated by work of many people, including some investigators at Stanford, where there's a very clear hierarchy. The mesenchymal lineage is actually kind of messy, and you'll hear people talking about mesenchymal stem cells, and yet we don't really have a good sense of what a mesenchymal stem cell is and what the differentiation pathway that those cells follow is. But so with Ewing sarcoma, it's an interesting tumor because it was initially thought actually not to be of mesenchymal origin. It was thought to be a neural crest-derived tumor, basically based on the expression of some neural crest cell markers in Ewing sarcoma cell lines. And also the propensity of these cells to form dendrites and express neural proteins when differentiation agents are added to these cell lines. So based on the expression of these markers, it had been thought that Ewing sarcoma was neural crest-derived tumor, even though it occurs in bone. That perhaps it was some kind of a neural crest progenitor that had migrated to bone. But the first indication that might not be the case was a study where somebody took a rhabdomyosarcoma cell line, which is obviously not neural crest-derived, and expressed the EWS-fly oncoprotein in those cells, and saw that there was expression of these neural crest cell markers, suggesting that actually the expression of the markers had more to do with the expression of the initiating oncogenic event rather than the cell of origin. So that was the first indication that perhaps that wasn't the cell of origin. And then right around the same time, a group in Italy showed that you could actually take these so-called mesenchymal stem cells or mesenchymal precursors from mice and express the EWS-fly and get a tumor that looked a lot like a Ewing sarcoma. So suggesting that perhaps some cell within the mesenchymal progenitor lineage was actually the cell of origin. And then more recently, using microarray profiles, another group has shown that if you knock down expression of the EWS-fly oncoprotein in cell lines, you actually get an expression profile that is clusters together with the expression profile of mesenchymal stem cells, which again are this not very well defined cell type, but in any case suggesting that again it's probably some type of mesenchymal progenitor that gives rise to these tumors and not the neural crest as had been initially thought. And this is kind of an interesting idea, sort of compelling concept going back to what Linda Boxer was talking about this morning. So if you take these cells where you've knocked down EWS-fly and put them into adipocytic differentiation media, you can actually get these Ewing cell lines to differentiate into adipocytes. So suggesting that if perhaps there was a way to combine blockage of EWS-fly function with a differentiation agent, we could have a similar blockbuster drug for Ewing's as retinoic acid has been for APML. So that's definitely a very active avenue of investigation, something that we're very interested in the lab. So again, going back to this concept of what are these mesenchymal stem cells that people work with, and as many of you have worked in this field, you realize it's a very murky concept, and there's some people that argue that there's a progenitor, a mesenchymal progenitor that gives rise to fat cells, cartilage, and bone, and can also give rise to muscle and neural cells, and some people, that's controversial data, and it's, some people argue that that may not exist, and then maybe there's a connective tissue precursor that gives rise to bone, fat, and cartilage, but it's a different precursor than the one that gives rise to muscle and neurons. So it's unclear, but one of the best papers that I've seen recently sort of clarifying this issue was a paper from an Italian group where they showed that CD146 positive mesenchymal stem cells actually have this unique capability in vivo, which is that if you transplant them into a mouse, they will form basically a bone marrow niche. So they'll actually form bone, they'll deposit bone, but more importantly, they will attract the bone, the mouse-derived hematopoietic cells to migrate to this area and basically form what looks like bone marrow, suggesting that these CD146 positive cells are an earlier mesenchymal progenitor that has the ability to form this bone marrow niche. So we were very interested in the idea that maybe, since this is a very early progenitor, maybe there was some relationship between osteosarcoma, the cell of origin of osteosarcoma, and these CD146 positive cells, or perhaps the cell of origin of Ewing sarcoma. So actually, I'll skip this slide and go back to it in a second. I'll get to the issue of the CD146 again, but this is just to show that these mesenchymal stem cells are typically characterized by their ability to differentiate into bone, as shown here in vitro, or also fat, and they have certain cell surface characteristics. But I wanted to point out that this isn't really truly a stem cell assay, because these are in vitro assays, and you don't really know for sure that these cells that are making bone are the same cells that can also make fat, or whether you have a mixed population, which is why that CD146 study was very nice, because it actually provided a true transplantation stem cell assay where you could show that it was these CD146 positive cells that had the ability to deform the niche in vivo. So we've been very interested in this concept of cell of origin, and the relationship between cell of origin and cancer stem cells. This is just an example of a study that's currently ongoing in the lab. Again, going back to Ewing sarcoma. As I mentioned at the beginning, Ewing's and osteo are both tumors that occur in adolescence. So we've been interested to ask whether mesenchymal stem cells isolated from adolescent patients or pediatric patients actually behave differently from mesenchymal stem cells isolated from adults in terms of how susceptible they are to transformation by EWS-fly as the initiating step in Ewing sarcomagenesis, and whether that might teach us something about why it is that these cancers seem to only occur in a certain age population. So that's something that's, again, work that's ongoing in the lab. And I want to just spend the last part of the talk talking about a clinical study, or really translational study, to try to study or identify some markers of aggressiveness of osteosarcoma by looking at primary biopsy samples. So as I mentioned before at the beginning of the talk, we have access to these samples at the time of diagnosis. The patient receives chemotherapy, and then there's a surgical resection, which we also have access to. So this provides us the opportunity to really compare what's different between this initial sample and the cells that actually survived chemotherapy. And we can look at patients that had very good response versus those that didn't, and try to see if there are differences in the two samples. So the way we've designed the study, so in order to expand the amount of tissue available to us, we started the study about a year ago, where we take the diagnostic sample and we actually transplant it into mice. It took us a long time to figure out where the best site to get these tumors to grow would be, but it turns out that they grow best in the subrenal capsule, so that's where we implant. So we take these samples from patients diagnosed with or they have radiologic features suggesting of either an osteosarcoma or a Ewing sarcoma, transplant them into mice, And then we follow those patients so that we know when they're going to get their surgical resection, and we have a sort of a pretty tight communication with the surgeons on this, and so they let us know when the patient's going to go back to the operating room. We going to go back in and get another sample, bring it back to the lab, and so the idea is to expand these initial samples in mice, and then compare the pre and the post chemo, looking for two things primarily. One is differential expression of surface markers. So as I'm sure you've heard from other talks during this meeting and before, there's this concept of the cancer stem cell, which is the cancer is a heterogeneous population of cells, and that those heterogeneous populations can be identified by surface markers. So one of the things we've been interested in is whether there's a relationship between this cancer stem cell and osteosarcoma and chemotherapy resistance. So perhaps the cells that are more stem-like actually are more resistant, so we might see an enrichment of the surface. We might be able to identify what those cells are by looking for differential enrichment of surface markers pre and post chemotherapy, and then obviously also looking for gene expression profiles to compare pre and post chemo samples. So these kinds of studies are never easy to do. It takes a lot of coordination, and so I just wanted to point out that it's oftentimes, particularly people who are not involved in clinical work, don't understand why we don't have access to sort of all of these samples. Dr. Fisher was pointing out earlier that with colon cancer, it's actually not very common to have access to samples metastatic tissue because there's an initial diagnostic biopsy, and then the patient's just assumed to have metastasis based on the clinical presentation. So it's actually somewhat unique that we have actually access to both these pre and post chemo samples. So these are just the samples that we've been able to collect so far. This is about a year's worth of work, because again, as I mentioned, this is a rare disease, so it takes a lot of coordination to get these samples. These are just some samples. We have the post chemo only and not the pre chemo. Some we have the pre chemo but not the post chemo, again because of logistical issues. So we're very excited actually about this one pair, because this is the first pair of sample that we got from the same patient, where we have the pre-chemo and the post-chemo, and this is actually a patient where there was a poor response to chemotherapy, which is probably why we were able to get the post-chemo sample. So we're very excited about following these. Some of these we actually have tumors that have grown in the xenografts in mice, and most of them we still have under observation. So this is kind of a work in progress, very early preliminary data. But I just wanted to point out that we can't graft these tumors, we can't grow them, and even though they look distinct from the primary patient sample, the really diagnostic aspect of an osteosarcoma is the ability of the tumor to form malignant or osteoid, or deposit this kind of abnormal bone. And we were very happy to see that in the xenografts we see deposition of this osteoid. So at least we know that some of the characteristics seen in the initial patient sample are being maintained in these grafts. This is getting back to something that I talked about earlier, this question of the cell of origin of osteosarcoma. We had looked to see whether both Ewings and osteos expressed this very early mesenchymal marker CD146, which has the ability to form these bone marrow niches. And we were excited to see that actually all the Ewings, don't have that here, but all the Ewings were negative for CD146, but the osteos pretty much uniformly are positive, both in the tumor samples that we've isolated, as well as cell lines that we've looked at have a very high expression of the CD146, whereas other fibroblast-like cells, for example, BJ fibroblasts, don't express CD146. So this is interesting for a couple of reasons. One, it allows us to potentially identify a marker of true osteosarcoma cells in these biopsy samples, where we know we have a mixed population of cells, including stroma and endothelial cells and other tissues. So if CD146 actually does turn out to be a true marker of the osteosarcoma tumor cells, that'll be very interesting. But it also suggests something about what the cell of origin of these tumors might be, and suggests that they may be derived from a very early mesenchymal progenitor, and that potentially trying to understand what are the self-renewal pathways that maintain those very early progenitors may lead us to some novel insights into osteosarcoma biology. And then again, getting back to this issue of comparing the pre and the post-chemo sample, this is very preliminary work, but I want to just put it up here as an example of what we're doing. So we're collaborating with Irv Weissman's group to try to identify surface markers on these CD146 positive pre-chemo versus post-chemo osteosarcoma cells, to try to identify markers that are differentially expressed, either enriched or depleted after chemotherapy, because with the hypothesis that if cancer stem cells are actually important in osteosarcoma, one of the reasons why they might be important is that they may be a reservoir for tumor cells that are relatively chemotherapy resistant. So if we can identify either enrichment or depletion of some of these surface markers, we can then go back and ask whether in the diagnostic samples, the cells that are enriched for CD71 in this case, actually CD71 is depleted, but CD117, which is a c-KIT, seems to be enriched in the post-chemo samples. CD51, 61 also seems to be enriched, and we have several other markers that we're also evaluating. So we can now go back to the initial biopsy samples and see whether samples that are CD51, 61 or CD117 positive have an increased ability to transplant to tumors and may potentially represent the cancer stem cell in this disease. So I just wanted to end the talk sort of moving to a slightly different topic, which is whether it's possible to model these cancers in mice. And the reason that I think that might be important is because, as I mentioned at the beginning, sarcomas, pediatric sarcomas, or pediatric cancers in general, are rare diseases. So whereas people have tended to focus on developing models in mice for the prevalent diseases like lung cancer and pancreatic cancer, and obviously there's a role for them. We have access to lots and lots of patients who have lung cancer and pancreatic cancer, so perhaps we don't need these preclinical models so much. But in pediatric cancers, which are very rare, the ability to do a lot of these preclinical studies in mice that closely recapitulate the human disease is another tool that may allow us to move further along in the goals of improving therapy for these patients. So this is from a recently published paper from the Stu Orkin's group, where they were able to show that by crossing mice that are p53 deficient and mice that are RB deficient, these are conditional mice where RB loss and p53 loss are mediated by expression of Cre recombinase, and they cross those mice with mice that express Cre recombinase in an early osteoblastic progenitor, and this again goes back to the question of what really is the cell of origin of these tumors. So they argue that the cell of origin has to be an early osteoblastic cell, because Osterix Cre, which is the Cre that is driving loss of p53 and RB in this model, only when they crossed the mice to Osterix Cre were they able to get tumors that were similar to human osteosarcomas, and when they used later Cre lines or earlier Cre lines, they didn't get these tumors. So this may turn out to be true, although we feel based on our human data that it's probably more likely that the true cell of origin is an earlier progenitor. So that might reflect the difference between mouse and humans. But nevertheless, this is a nice model. It provides obviously more material to look at potential preclinical therapies that might then be selected for the ones that would be most appropriate for the small numbers of patients that we do have. And another interesting aspect of that paper was that they actually looked at lots of different gene expression profiles across many different sarcomas, both in mice and in humans. And what they showed is that the clustering actually went across species. So osteosarcomas from mice clustered together with osteosarcomas from humans. So these red boxes are the mouse osteosarcomas, and the sort of pink circles are human osteosarcomas. So they're kind of clustering together, which is pretty exciting, because it suggests that even though obviously mice and humans are very different, that the expression profiles of these tumors across species are very similar, suggesting that these models actually do closely recapitulate the human disease. So we have some work in our lab now trying to build a similar model for Ewing sarcoma, as what I described here for osteo. So I just wanted to conclude by again saying that both Ewing's and osteosarcoma are bone sarcomas that occur in a similar location in patients, but they have a very distinct biology. The cell of origin for both of these cancers is not well defined. The future of treatment for both Ewing's and osteosarcoma will require better biology-based therapies. We need to also, as I mentioned at the beginning, there's been some big failures in some attempts to find new biological therapies that probably mean that we need better preclinical models, either dog models or mouse models, to really design these trials better and identify those therapies that are most likely to work in human patients. We need to have a better understanding of the cell of origin and the key pathways that are required for survival in order to understand how to design biological therapies for these cancers. And in this respect, mouse models may be particularly useful for these rare diseases. So, oops, well, I missed my acknowledgement slide. I just wanted to acknowledge the last part of the talk, the work that I discussed from our lab, been carried out by Tom Johnson, who's a very talented postdoc, and recently also assisted by Jin Chang, who's in the audience, who's a PhD Hemong fellow, who's doing great work in the lab, and the rest of my group that I didn't get to mention. But thanks for your attention, and happy to answer any questions.
For more, please visit us at stanford.edu.